Hello, everybody. Thank you for joining us for another story time this week. I'm super excited about this week's theme. If you're part of our daily Facebook activity group, the theme this week is all about invertebrates, and we'll talk about what that means in just a minute. But before we do that, we're going to talk about we have 6 major groups of animals, and I want you to try to guess what they are. I'll give you a hint. I just told you what one was with our theme. So that one is invertebrates. And our invertebrates are the opposite of our vertebrates. So if you feel down your back, right, you can feel that backbone running down the center of your back. So any animal with a backbone, we call them vertebrates. Any animal without a backbone, we call invertebrates. So that's one group, and we'll talk more about them. Those 6 groups are invertebrates, mammals, birds, fish, reptiles, and amphibians. Okay, got all that? Great. All right, so then let's talk about our animals without backbones. Probably the most well-known group is our arthropods, and those things all have exoskeletons and jointed legs. Who might fit into that category? If you said insects or spiders, you are correct. Also think about our crustaceans, so things like crabs or lobsters would also fit into that category. Another group of our invertebrates are our mollusks. So those are squishy body things. Some of them have a shell, some of them have a foot, some of them have tentacles. So think of things like snails and squids. And then we have a different category for some other squishy body things that also have stinging cells. So those would be our jellyfish, and that group is called the cnidarians. Then we also have a group called porifera, which are the sponges, mostly in the ocean. And we're missing one. Who are we? Oh, we're missing a couple. We have our annelids, which are our worms. We have our echinoderms, which are hard-bodied things like starfish. Okay? So those are all of our animals without backbones, and they all those categories have super cool ways of surviving and getting what they need from the environment without their backbones. But today we're going to be talking mostly about insects because they are the kind that we see the most of where we live. So instead of having bones on the inside, our insects have their bones on the outside in what we call the exoskeleton. And we're going to read a story today. It's called Buzzing with Questions, The Inquisitive Mind of Charles Henry Turner. And this is what we would call a biography, so it tells us about the life of somebody who contributed a lot to the knowledge that we have about insects today. So he had a lot of questions and he found a lot of answers, and we're going to read today about what he now teaches us about bugs. So, it was written by Janice Harrington and illustrated by Theodore Taylor. Buzzing with Questions, The Inquisitive Mind of Charles Henry Turner. The study of biology trains the powers of observation. I wholeheartedly believe in this quote. All right, here we go. Questions that itched like mosquito bites. Questions that tickled like spider webs. Questions you just couldn't shoo away. Questions hopped through Charlie Henry Turner's mind like grasshoppers. His brain buzzed with questions about plants and animals and bugs. I can relate. His parents' home swarmed with books, but it was never enough books to answer all of his questions. Charles Henry Turner asked so many questions that his teacher urged him to go and find out. And Charles did. Sometimes books might not have your answers. You're just going to have to observe with your eyes. He read and studied and he worked hard. And after he finished high school, he did what many people thought impossible. Even though he was a janitor's son, even though most colleges didn't accept African American students, even though it meant more hard work, Charles Henry Turner went to college. So this was quite a long time ago, when colleges didn't accept African Americans. So what he did was pretty remarkable. In his biology class, Charles met the magnetic teacher, Clarence L. Herrick. Herrick's classes hummed with energy. Students chatting, students examining the organs of small animals, and students staring one-eyed through microscopes. On Friday afternoons, Herrick invited students to his laboratory for spirited talks about biology. He spread tablecloths over the long laboratory tables and set out sweet cakes and cups of tea. But Herrick worried about inviting Charles Henry Turner. He worried the other students wouldn't want to drink tea with his only brown-skinned student. How do you think that made Charles Henry feel? To Herrick's delight, the other students wanted Charles to join them. They liked the shy, quiet student who always earned high grades, the hard-working Charles Henry Turner. Charles was indefatigable, a classmate said. He spent hours peering through microscopes, planning experiments, gathering specimens, keeping records, drawing charts, and reading scientific papers in French and German. I don't know French or German. But whatever the language, he never stopped asking questions. One question led Charles to a small, eight-legged, eight-eyed, two-fanged creature, the spider. Charles wanted to know if spiders could learn or if they were only weaving machines that made the same web over and over. Charles searched for spider webs. He trudged through meadows, inspected stone walls, and scouted the sides of railroad tracks. He toppled wood piles, lingered over logs, and peeked into dusty corners. Charles found webs. Lots of them. He even spotted double webs. Well, that would be cool. That looked, he wrote, as if the spider were trying to fish with two lines instead of one. All kinds of spiders, all kinds of webs caught Charles's attention, even a web between a windowsill and a wall. What would the spider do, Charles wondered, if he swept away its web? With a broom, Charles brushed away the web. Not knowing it was part of a science experiment, the unsuspecting spider rebuilt its web. Sweep away, rebuild, sweep away, rebuild. After losing its web for the 5th time, the spider gave up and wove a new home beneath the windowsill. But Charles didn't give up. He repeated his experiment with arachnarium, a spider jar. Never heard that word before. It's super cool. He filled the bottom of the jar with sand and pushed a post into the sand. Then he added a spider. He slipped a paper triangle into the jar and watched the spider. Later, he removed the triangle and replaced it with an L-shaped tube and watched again. With each change, the spider rewove and reshaped its web. Charles concluded that instinct told spiders to make their webs, but that each spider wove a web just right for its home. Charles called this intelligent action. Spiders were not just weaving machines. So he's proving that spiders are observing their surroundings and building the best possible web that they can for where they're building, which is something we never knew. And that's really cool. The indefatigable scientist then wondered about even smaller animals. In scummy ponds and weedy ditches, he searched for tiny crustaceans. He found seed shrimp, water fleas, and wheel animals. Through his microscope, he admired their small bodies. They were beautiful and translucent, and some looked like a nest of tubes. Charles even discovered a new crustacean and named it Cyprus herricki, after his friend and teacher, Clarence L. Herrick. So he's saying these are crustaceans, so these would be arthropods, which is one group of our invertebrates. But Charles wouldn't stop asking questions. At 39, he returned to school. He wanted to learn. He wanted to ask big questions about other small creatures. The ant. Charles wondered how ants found their way home. Did ants have a hidden power in their brains that pulled them to their nests? Did the sun guide them? Did they follow a trail of smells? Have any of you ever wondered this question? I have. Searching for answers over the next 5 years, the scientists studied acrobat ants, big-headed ants, false honey ants, and odorous house ants, all kinds of ants. To tell one ant from another, Charles marked their abdomens with watercolors. He built an obstacle course with cardboard stages, cardboard rooms, and cardboard ramps. Ants crawled over the stages. Ants scurried over the ramps. Ants tapped at the cardboard with their antennas. Ants searched for their nests, but Charles didn't help them. Instead, he moved the ramps to unexpected places. He angled ramps up and down. He slanted them. He angled ramps up and he slanted them down. He painted ramps different colors. He shifted the light and the direction of the light. He smeared stinky oils over the cardboard paths. He caused his ants trouble. But by watching ants find new ways to reach their nests, Charles learned how they solved the problems. Those poor ants, it's quite the obstacle course. Charles drew a map with squiggly lines and arrows to show the wandering path of a lost ant. A French scientist called this wandering tournement de Turner, or Turner circling, in honor of Charles Henry Turner, the scientist who taught us that ants were not mindless robots. Each ant used sight, sound, touch, smell, and movement of its body in sunlight to find its way home. So that's a lot of things they're using. Ants must be pretty smart. Like the ants, Charles now searched for a new home. He found work as a biology teacher at the first African American high school west of the Mississippi, Sumner High School in St. Louis. Excited by their new teacher, students filled their lab books with notes about his class. One student wrote that Charles set out dishes of jam for bees at breakfast, lunch, and dinner. The bees circled and buzzed at each meal. But then, when Charles set out jam only at breakfast, the bees still invited themselves to lunch and dinner. Surprised, the students learned that even bees sense time. So they got so used to being fed at each mealtime that they just showed up at the right time hoping for food. Questions kept circling and buzzing in Charles's mind. In a St. Louis park, the scientist placed red cardboard circles in a patch of clover that swarmed with bees. He coated the circles with honey, and then he waited. The bees weren't interested in his circles. They weren't even drawn to the smell of honey. That seems weird. But good scientists are patient, so Charles tried again. He caught bees in a bottle and toppled them over the honey. At first, the bees buzzed away, but eventually they settled on the cardboard to enjoy a sip of honey. Once the bees learned that the red circles carried honey, Charles replaced the red circles with honey-filled blue circles. The bees, ignored them. Whether he used circles or cones or boxes or added new colors, the bees kept flying to the color red, even when another color held lots of honey. In 32 different experiments, no matter how Charles tried to trick them, the bees chose the color red. Charles Henry Turner was the first biologist to prove scientifically that bees could see color. So the red circles, the color was the only thing that made them different, and the bees only went to red. So then we know that they must be able to see that color. Now we know that bees not only see color, but they can see in UV light too, which is pretty cool. Bees, giant water bugs, whirligig beetles, dragonfly nymphs, water spiders, paper wasps, hornets, or tent caterpillars. Charles studied them all. He taught cockroaches to find their way through a maze, proving that they could learn. He triggered moths to beat their wings whenever they heard a whistle, just like Pavlov, a Russian scientist who trained dogs to drool whenever they heard a bell. He frightened doodlebugs, or antlions, which made them lie still. With a magnifying glass, he studied how they moved and trapped their food, how antlions grew, and everything else that antlions do. Charles learned so much that he became the world's first doodlebug expert. Doodlebugs are a real thing. Patiently, he watched 1,000 caterpillars crawl, slowly, up a vertical maze, learning, slowly, that caterpillars can find their way home by trial and error. Charles Henry Turner was indefatigable. So we've heard that word, indefatigable, a few times now. What do you guess that it means? What is Charles? So it means he's persistent or tireless, like he just keeps going and he wants to find out more. His mazes, spider jars, paper circles, and cardboard stages toppled old ideas about insects. He never stopped inventing new ways to study the smallest creatures, searching for new ideas or asking questions. Questions that itched like mosquito bites, questions that tickled like spider webs, questions that only a good experiment could shoo away. As hardworking as the ants and bees, he studied. Charles published over 50 scientific papers on everything from bird brains and the bathroom habits of cockroaches to blind crayfish and the growth of grape leaves. He was a pioneer scientist of animal behavior, an internationally admired entomologist, and one of the leading African American scientists of his time. But even though he was a respected scientist, Charles faced racial prejudice. He lived in the South, where African Americans had to attend separate schools and where they could rarely vote. He lived in St. Louis during the terrible East St. Louis riot, when hateful mobs killed more than 100 African Americans and burned their neighborhoods. Yet despite this prejudice, Charles wove himself into his community. He raised money for poor families and led a settlement house that sold lunches to hungry children for only a penny. He worked with white and black people to make St. Louis a better place for everybody. Biology, the study of plants and animals, gave Charles hope. He wrote that biology could help people see the connections among living things. Biology taught us to think less about ourselves and more about others. Charles Henry Turner, the boy who never stopped asking questions, grew into a tireless reader who owned 1,000 books. He was the good friend, always willing to help other scientists by offering ideas, helping with research, or sharing his equipment. If my micro camera proves a success, I shall be glad for you to use it whenever you desire. Charles Henry Turner, the boy who never stopped asking questions, grew into the determined biologist who would stay up all night to watch a spider or spend all day observing wasps beside the railroad track. I shall take my camera along and get a few photographs. His curiosity danced from experiment to experiment. It moved in Turner's circles, in Tournament de Turner, always exploring, always researching to discover new ideas. Charles Henry Turner, the boy whose teacher urged him to go out and find out, grew into a teacher himself, a devoted scholar who taught students to look closely, to find the webs that connect us all, and just as he did, to fill the world with questions, questions, questions. And I won't read everything about him, but one of the quotes here, it says, "Often I have failed, my patience not being a match for the persistence of the ant. In other cases, by patient persistence, I have succeeded." Okay. So, this is Charles Henry Turner. He's one of the people I look up to because of the way he approached everything and asked questions and didn't do things the same way that people had done them before. He was kind of a pioneer, not only for African Americans, but for people studying science and coming up with these new crazy experiments and ways to prove his ideas or predictions if they were right or wrong. And that's super cool. So I can't really go out on a walk in nature without thinking of a billion questions, just like Charles Henry Turner did. And I hope that when you're out walking in nature this week, or forever really, that you're asking questions about what's around you, and hopefully you're inspired to try to learn more about it, just like this guy. If you're part of our daily Facebook activities group, we'll be posting the schedule so you can see what's coming up and which invertebrates we're going to be talking about. But otherwise, I hope you all have a fantastic week exploring and learning in nature. We will see you next week for another story time. Bye!